Okay, great, thank you. I'd like to thank the organizers for inviting me and organizing this pretty amazing mix of speakers and students and researchers. Certainly been very stimulating. What I'll do today is try to give you a taste for some of the ways in which people are trying to apply Bayesian methods to understand the brain. And I guess I could say feel free to interrupt me anytime, but from the I don't probably have to say that because it's been happening all the talk, so I'll just leave it up to you. So why don't we just begin with the man, right? So Thomas Bayes. There's been a controversy, which is why I put that question mark there, right? But who was this person? So some of you might already know that from his appearance and dress, he was a non-conformist minister. So one of the first publications he had was this one, Divine Benevolence, or an attempt to prove that the principal end of God, basically, is the happiness of his creatures. So pretty noble attempt to prove that God is kind and benevolent, right? And that's the publication on the right side. You can probably download that from Google search. I don't know how many citations it has, probably not as much as his other publications, but the other publication he had, this was before he died, was an introduction to the doctrine of fluxions. Anybody know what are fluxions or fluxions? Yeah, so basically Newton's differential calculus. So this was an attempt from Bayes to basically show that this particular theory of Newton's was really correct, mathematically and logically correct. And I guess this was after this big controversy with Leibniz and Newton and so on. So basically this was a contribution to that literature. And finally, of course, the one that he's recognized for is his posthumously published essay on solving this problem in the doctrine of chances, where he informally defined Bayes' rule in a way. So why is this really interesting from the point of view of people working in cognitive science and AI, and robotics and so on, and in computer science also? Well, it gives you a way, as Nazar pointed out, it gives you a principled way of combining prior knowledge with information coming from sensors, data basically, right? So I'm calling P of X belief. That's another word people use. And so I think it sort of personifies this notion of probability as being something that you hold as a belief. And P of X is what you might believe in before you encounter some new information. I'm calling that I. And Bayes' theorem, as you saw in the previous speaker's lectures, it gives you a way of combining prior knowledge with new information, and so that's just the revised belief that you have. So you can call this a revision of your prior belief. And so before you had P of X as your belief, and now you have P of X given this new information. And you could use semicolon if you don't like the slash.
Depends on whether you're observing it or assuming.
Right, exactly, yeah. So that was something that was mentioned by you in the previous slide, in the previous talk. So to motivate this, what about this image here, right? So I'm going to try to sort of convince you that we're already doing Bayesian inference whenever we do perception, right? So who is this man? So those of you who know who this person is, by just looking at this person's fingers, raise their hands. Oh, okay, so this is going to be a pretty easy, yeah, that's right. So if it's to a bunch of scientists, it's going to be pretty obvious, right? So, but I'll also take you through the examples. I'll give you some more sensory information, this eye that I called you, so giving you some information about the hair. So how many now? Anybody who didn't? Now they can recognize it? Okay. Anybody who still can't recognize the person? Oh, there's still some person. Cool. So we can still do the experiment. So I'll give you some more information. Hopefully your posterior probability of who this person is going to increase among all the other possibilities you have. So now, that should be pretty obvious, right? And finally, of course, the mustache is pretty unmistakable, right? So most of you probably already guessed it's Albert Einstein, right?
Can I make a comment? I guessed it in my mind. Declaring through his sweater.
Okay, there we go. That's right. So it doesn't have to be just one piece of information. It could be any sort of piece of information from the given information you have that sort of triggers a particular response, right? And that you can use to integrate to get your posterior belief. So here's the object of study of many scientists, neuroscientists, and cognitive scientists, which is a 3-pound universe. So forget about what the physicists tell you. The universe is not out there; it's in here, right? And this is the universe we live in, right? It's only 3 pounds. It's made of squishy biological tissue, and it's still a big open question, right? One of the big fundamental scientific challenges of this new century is figuring out how this organism, or how this machine works, right? So how does this squishy piece of tissue work? And so there's a growing sort of sense that perhaps one can use Bayesian methods to understand at least the data that's been there for a while and also the new data that's coming in, both from behavior as well as from recording from the brain in animals and in some cases humans. And so I've been involved in some of this effort. There's a couple of books that I edited with other researchers. And what I'll do today is just give you one example of the kind of research that we can do. Part one is going to be today, it's going to look at an example of how you can start to think about implementing. So if the brain really is Bayesian, how is it actually using the biological cells to implement this mathematical concept called Bayesian inference? Obviously, it's not going to be an exact one-to-one match, but maybe it's approximating Bayesian inference in a particular way. So let's see how we can try to understand the link between the biology and the mathematics, right? Is there a way to connect them together? And in tomorrow's lecture, I'll try to connect this to this notion of rewards and actions. So once you have, let's say, a way to compute posterior probability distributions in networks of these brain cells or neurons, how can you then use it to make decisions and actions? And maybe there's a way to tie it into the rewards. So we know that the animals will work to maximize the rewards and so on in many circumstances. So how do you use probability distributions and rewards to figure out what actions to take? And that's something I'll talk about tomorrow. Okay? And so here's the bigger question. So how does the brain parse or interpret noisy, ambiguous sensory inputs to generate actions? So that's sort of the big goal, is combining sensory information and prior knowledge you have, combining that to compute these posteriors to figure out what actions to take. And the hypothesis that a lot of people are looking at is that perhaps it's some form of approximate Bayesian inference that lets you do that. So let's start with another example. So how many of you have seen this image before? Okay, so that's better, a lot fewer. So do those who have not seen it, can you look at it for a while and tell me what it is? It's Albert Einstein, okay. So there's a prior bias that I introduced, right, in the previous example that it could be Einstein, right? But if I give you a clue, most of you will probably be able to figure out what it is, right? So what I'm going to do is give you some top-down bias, is how the psychologists call it. So I'm going to point you to a particular location, and I'm going to give you the semantic cue in terms of language, and that'll let your brain sort of increase the posterior probability of one particular interpretation. Oh, that's a good auditory. So what he suggested was a dog, right? So anybody, if I just tell you dog, can you see a dog there? Yeah? If I say a Dalmatian dog, is that helpful? Okay, I'm going to point you. So there's the Dalmatian dog sniffing down here. So here's the head, there's the leg. How about now? Many more? You can see it, right? You can see it. So once you actually have that interpretation, it becomes almost impossible not to see it, right? I mean, that's what you see when you look at this image. So it's a very noisy, ambiguous image, but then once I tell you what it is, that there's a picture of a dog there, and there's a leg, and there's another leg, and so on the body. There, that you can start to see it, right? So one way of looking at that is to say, well, initially maybe you had some prior distribution over what could be. So, if you thought it was a street, maybe you might have thought even it was a dog, but you weren't really sure. Pondicherry Beach, maybe you had a couple of drinks and you're wandering around the beach, the promenade. So then you maybe have some belief that maybe this could be the beach, right? That might be the belief before you saw any information, but then if I give you some more information, such as this particular image, then you might have this kind of a likelihood function, right? So things that it might say that each of those possibilities have some chance of generating this kind of an image. And then the posterior probability would give you something like that, which is, before I gave you the clue, many of you had that kind of a distribution potentially, right? So you were not certain of what the interpretation was. And so when you, for example, make a decision, which is sort of sampling from the distribution, or picking the maximum value, you might pick any one of those possible interpretations. You weren't sure which one it was, right? But if I gave you the clue as to look at that particular location, then maybe it helped to increase your prior probability for a dog, and so then when you combine that with the likelihood, you might have a higher possibility of seeing that particular interpretation, right? So the decision itself would be biased towards that particular interpretation.
What's the corner of this one?
What's that?
What's the corner of this?
What's the corner?
Yes.
You mean over here on the left side?
Is that a tree?
Yeah, that's supposed to be a tree, I think. It's basically a tree, the trunk of a tree, right? Here's another example. So this is a very classical example from computer vision, or I guess vision. So in vision, right, you have a retina, and then you're projecting a three-dimensional world into a two-dimensional image, right? So what do you expect when you have three-dimensional objects projected onto a two-dimensional image? You're going to have ambiguity, right? So I'm going to use a prop that I actually took from the tea session that we had, right? So basically one of these. Maybe I'll take it as a souvenir. Don't tell me. Just kidding. So you have this particular object, right? So if I tilt it slightly at 45 degrees, this way or this way, right? The projection on your retina is going to be this oval image, right? Because it's tilted now. And so if I give you just that image, you can't really tell me what it is. It could have been a circular object that you're looking at from a different orientation, like 45-degree orientation, or it could actually be an oval object, right? So maybe it's an oval plate, right, that you're seeing. So which one of those interpretations is the one that you actually think is on the retina, right? So obviously that's a problem of ambiguity, right? So one way of solving this problem, which was suggested by Geisler and Kirsten back in 2002, is to sort of looking at it from a Bayesian approach, right? So looking at it using a Bayesian model. So you can look at two different aspects. One is the slant in terms of how the viewing angle, right? The other is the aspect ratio, which is, you know what the aspect ratio is? The width divided by the height of the circle, right? So the circular object. So it could be oval versus circular and so on. So if you look at that, you can compute this likelihood function, which is...
But you have the option to view a different angle, right?
Yeah, so basically this particular animal or robot, right, it doesn't really know what angle it's looking at. So for different angles, right, for different angles and different aspect ratios of the object, you're going to have this kind of a likelihood function, which basically says, for example, that if the aspect ratio is 1, which is a circular object, then there's two possibilities of high probability. One is that you're 45 degrees minus 45, and the other is plus 45 degrees.
I think to answer Haritha's thing, I think the point is you're not viewing the object, you're viewing a photograph of the object.
Yeah, that's right, yeah, exactly. So you're looking at the image, so I is the image. So what is the probability of getting this particular image on your retina, given that you have two different parameters there? One is the slant, the other is the aspect ratio. Right? So those are the two different things that are changing. So as you change those two aspects, these are this particular image is generated. Right? And that's the probability of that particular image being generated. So given this particular likelihood function, then now you can apply Bayes' theorem to figure out the posterior, right? So the important question is, what about the prior, right? So what is so that's the critical part, right? Yeah.
It's just a technical question. Isn't this a deterministic situation? I mean, it's just ray optics which will tell you what where's the probability here?
Yeah, I mean, I think ultimately you have to assume that the sensors are noisy, and so there's some amount of, I mean, it's basically that line that is the deterministic part, right? That's going to be, and then there's some variance around that line, but you're right. I mean, in a purely sort of, maybe in a man-made system, you can make it so good that it's all just you know, the line, but then in a biological system, there's always going to be noise, and so there's some amount of perturbation that, you know, and this is supposed to be a function that the system has learned, right? So it's learned through just experience, so there's always going to be some amount of noise. And so here's the prior probability that one could assume. So this assumes that the animal is typically experienced things that are, you know, almost circular, and then they're always sort of at that particular pi by 4 angle. So maybe it's an animal that lives always in a particular environment where it always tends to see these objects that are, you know, at that particular angle. And so there's that could be changed, right? Depending on how you set this prior, when you multiply those two, you're going to get a posterior probability that's over here. That looks like this, like the yellow shape distribution. It's slightly off, but it's heavily biased towards the prior probability. Right? So this just gives you one example of how you could start to disambiguate these kinds of problems, and these are called ill-posed problems in computer vision. So these ill-posed problems can be solved if you have appropriate prior probability distribution. Yeah.
But I thought that the animal doesn't know what the actual aspect ratio is. It only sees some image, but it doesn't.
Yeah. Yeah, I mean this is the distribution over all possible aspect ratios, right? So that's the part it's presumably learned through experience or through the genetic code, right? So it has to have some knowledge of these distributions, both the prior as well as the likelihood, and then it can combine them to figure out for this particular image that it's seeing right now, what is the interpretation that's most likely or most the maximum posteriority interpretation. So the dot is basically the maximum posterior probability interpretation, which is that there's something that's in this aspect ratio at that particular slant, right?
So you're thinking the prior is something that's evolved over...
Yeah, that's a good question, yeah. That's sort of the, so let me get to this slide here, which sort of talks about that, right? So where does the prior comes from? So there's a ton of experiments people have done with humans and also with animals that have to do with things like motion from cast shadows, surface perception, 3D from 2D, color perception, all of these. And what they've shown is that the brain appears to have evolved a set of priors that you can then use to interpret these ambiguous or noisy images.
So
and so, yeah.
Is there a direct access we have to the prior? Is it sort of a measurable quantity?
Well, you have to infer it from the data.
So,
right?
so I'm wondering then, if that's the case,
yeah.
Can you distinguish between the brain not doing a Bayesian thing, doing something else, doing some crazy thing, or having a prior and doing a Bayesian? Can you distinguish these?
Well, so there's always going to be alternate models that can also explain the data, but the beauty of the Bayesian approach is that it seems to encompass a whole bunch of data that you would require individual models to fit otherwise, right? So, for example, this paper by Weiss et al., the motion illusions, they showed that you can explain a whole bunch of illusions that people have used individual, very specialized models to explain before. But now if you make just a couple of simple assumptions that, in their case, it's that the brain is tuned towards, it always assumes that motions are smooth, that they're not just like jumping around, objects not jumping around, and also that the motion has a peak around 0 velocities, so that things tend to move slowly in the natural world. If you make those two assumptions, which are ecologically quite valid, because that's what happens in the natural world, then you can explain a whole bunch of illusions. So you might have heard of the barber pole illusion, where this barber pole moves up, but then things seem to be going down. Or the spokes illusion, so the cycle spokes are moving in one direction, but it looks like it's going in the opposite direction. So why should that happen, right? That's not really, it's happening because of those two assumptions. The prior probability the brain is using for interpreting is basically that things are going slowly and things are going smoothly. So if you use those two, then you
are
can
these
get
priors universal?
universal in what sense?
All the individuals have that.
Yeah, so at least in the subjects they've tested, it seems to explain their data quite well. So at least for those subjects.
Everybody has the same prior.
Oh, I guess, I mean, that's one of the assumptions you'd make. If its brain is evolved ecologically.
I guess you can change your priors if you spend your life staring at weird optical illusions.
That's true. That's true. Here's one other, I think this is an interesting, cool illusion, or I guess experiment that you can do, that we can do actually right now, which is, you can actually dramatically change your perception of object motion through just changing the shadow. Okay, so here's a ball, and here's a shadow of the ball, and the ball is going to move in the same way in both cases. There's going to be two cases, but the way it moves is going to look very differently depending on how I change that shadow that's cast. Right. So here's the first motion. Do you see how it changes? So the ball is going exactly the same way, but your brain thinks the ball is going in the three-dimensional way in the second, right? So how can you explain this? I mean, you could explain it by saying that there's some weird illumination that's changing also, right? But the assumption that really explains this is that the brain is assuming there's a single stationary source of illumination that is at the top, that's not moving along with the object, because I guess, presumably, ecologically, the sun is being the source of illumination for animals, and it doesn't move dramatically. So if you assume those kinds of things, then you can explain these types of illusions.
That is just a comment. I mean,
Yeah.
this could also be nurtured other than nature. So, you might have the neurological circuit.
Yeah.
And it doesn't have to be pre-coded at the genetic level.
So there's actually some results from the 1960s that show that if you take kittens, kittens are born with some amount of selectivity for different oriented bars and so on. But if you then grow them in an environment that is only has walls that are vertically striped, then they lose the sensitivity to other directions. And so they won't recognize motion that are like in opposite directions and so on. So you can change that. There's a critical period of about 10 days, at least for cats, and probably for humans also, where you can change the biases that are built in by evolution, and then that will help you process information in the future.
So I believe there are very low-level ways. I mean, there are individual neurons that fire when you detect motion.
Yeah, I'll show you one of that. I'll get to that. So in the time that I have, I mean, if I go over, you can stop me. Basically, I want to ask these two questions, right? So one is, can probability distributions really be represented by neurons? And secondly, how can you compute these posterior probabilities? Can you really do Bayesian inference, right? So here's the experiment that sort of answers your question, which is, how do you record cells in the brain, right? So this is an experiment done in the 1960s in cats. So you put a wire called an electrode into the brain. This is the visual area of the brain. And you show some stimulus. In this case, it's just bars that are moving in particular directions. And then what you get out of this is responses from the brain cells. So if you're close enough to a cell, you can listen in on the activity of the cell. The cells, basically, they emit a pulse. It's a digital output. So this is 0 or 1. So when the cell spikes, you're going to hear something that's called a spike. It's a pulse. So you hear tuck, 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 tuck. You hear those kinds of noises. And so what you'll see is, I'll put this.
So where are these?
These are cells in the visual cortex of the cat. So these are the back of your brain. So that's the first visual processing area of the brain.
So your retina maps first to this area.
That's right. Retina maps to one other area called the lateral geniculate nucleus, and from there it's going to this particular area. So it's only like a one step away from the retina actually. Okay, so here's the experiment done where the experimenter is moving. Can you hear it? So when they move in the other directions than this slanted direction, you don't hear any responses. It's like static, right? It's like Darth Vader's lightsaber, right? If you know Star Wars. This was done before Star Wars. This was the 1960s. But what you're hearing is the cell responding like static, so it goes current, right? It's basically when this bar is moved in that region of space given by the dots there. That's when you get the most response. But if you turn it in other angles, you won't get any response. The cell is very selective for orientation and the width of the bar and so on. So they're very selective responses. They're called orientation tuned, and the simple cell is just a main product.
So I guess my question was, is this selectivity at the single cell level innate or not?
Yeah, so that's that experiment I talked to you about. So some of it is innate, so when they're born, they already have some selectivity, but it can be changed by experience. Okay, so that's the, and so one of the observations is that the neuron responds by increasing its firing rate. So you can look at the number of pulses per second, and whenever you have something that the cell likes, it'll start to respond really highly. Okay, that's a very common property of neurons in the auditory, visual, somatosensory, and other systems, is that they all prefer selective parts of the world that they're processing, and then they'll fire really, they get really excited when you show them something that they like. And different neurons will like different kinds of stimuli.
Excuse me?
Yeah.
It can be like a cumulative response also, right? When you are going at an angle, all the neurons are coming at the same time.
Yeah, so you're talking about the mechanisms, how is it actually behaving and so on. So this actually shows you pictorially this response, right? So you can see each of those bars is a spike or one of these pulses. And so when you're orienting it in a vertical direction for this neuron, it responds the most. You have this rapid burst of spikes there. But in the other orientations, you only get a little bit, some sparse activity, right? So, and you can compute a tuning curve, which is basically showing you the orientation and the cell's response plotted there as sort of like a bell-shaped function. So what that's telling you is that this particular neuron likes vertical bars, but it will also respond slightly to nearby orientations.
I think the question was, is this neuron responding to vertical bars?
Yeah.
Is the vertical bar stimulating some group of neurons in a way that other bars don't, and therefore you get some...
Yeah, so I mean that's, there's models that are constructed that try to explain. So this is looking at one single cell, but it might be the emergent behavior of a whole bunch of neurons connected to it that are causing it to respond in this particular way. Right? So that's the mechanistic question. How is it actually generating this selectivity? Right? So there's multiple models that are explaining. So the one could be feed forward, the other could be recurrent, and so on. There's multiple ways of doing that.
So aren't these laid out in some sort of nearest neighbors?
Right. Yeah, so nearest neighbors tend to respond similarly and so on. So it's a very nice spatial pattern that you can look at, and so a lot of people have looked at that. But yeah, as you look closely, you'll get different kinds of orientations, but nearby ones respond similarly. So here's one of the issues, right? So if you have parameters such as theta, let's say the orientation of bars, that's a continuous set of parameters, right? It's a continuous space. And you might want to represent a probability distribution based on this. So maybe you have an image that contains a whole bunch of edges and so on. So how do you have a bunch of neurons represent this kind of a probability distribution? So one very naive way of doing that, which I mean we could start to use, is what if you just approximate this using a discrete approximation where each of the neurons has some particular preferred orientation, as we saw, right? It will respond the most to one particular orientation. And if you look at a population of these cells, then what you're going to get is different firing rates for those different neurons. And then you can basically decode the probability distribution by looking at the firing rates of each of those neurons, right? So you have a population of neurons, they're all responding in different ways, but if you look at the population, you can decode the underlying potentially probability distribution, right? So there's different algorithms for decoding and so on. The most naive one is to just literally say that that continuous space is discretized here, and then you're looking at the probability distribution as a discretized histogram, right, of the firing rates. So you can look at it that way. So you can then ask, how do you, given that you have that kind of a representation, how do you compute posterior probability? So you then have to answer the question, how do you model a network of neurons in the brain? So here's one of the very simple models that you can use, which is you have some input neurons, which are these circles. Each of them is supposed to represent one input neuron, and then you have the other circles, the ones at the top, and those are supposed to be, let's say, the output neurons. And then each of these connections is basically the connection from the input to the output, and also between themselves. And so you can use this matrix W to represent the feed-forward connections, the ones from the input to the ones that are called V there, and the R are the recurrent connections. Okay. I'm doing a time. Okay. So basically, these are the inputs. So imagine this as being the image that the cells are receiving, and these are the ones that are processing the information. And each of these are the connections between them. So you're assuming they're connected to each other completely, let's say, and that's connected given by a matrix R. And then the W matrix tells you the connections, the weights. These are just numbers, the positive or negative, between inputs to these intermediate layer neurons. And the dynamics is defined by this differential equation. It's a very simple linear differential equation. So you can look at this as the equations of motion, right, for the neurons. They're just defining how these cells evolve, their firing rate, as a function of both the inputs as well as the activity from the past, from the previous time step. And there's a time constant that defines how fast you can converge to each of these values that you're setting it to. Okay, so it's one of the simplest kind of ways in which you can model a recurrent, feedback set of neurons in a cortical area.
If it is of the past implies all the previous experiences in relation.
Well, I mean, this is basically just, if you look at the discretized version, it'll be just the previous time. So this captures the activities from all the previous time steps also, right? Just the activity of the previous time step, right? It's a function of the activity in the previous time step and so on, right? So it sort of goes on until the beginning of time.
But this is just a differential equation where
Yeah.
t is t current.
Yeah, I mean, this is a differential equation that's, you can define it in this way for a discrete time implementation, and so then you have this kind of an equation where it tells you how the t+1 activity is related to the activity at t, as well as the activity that you got from the inputs, which is this it, right? And the w is basically the feed-forward weights, the weights that tell you how the input is modulating the activity here, and the r is telling you how the activities from the past time step are modulating the activities at the current time step. Okay, so there's two sets of weights, w and the r, and those are related to the matrix w and the matrix r there. Okay, so very simple. You can make it nonlinear, make it more complicated, and so on. But this is like a very simple set of equations. And the thing to note is that this particular model of biological activity, it has both this activity at one time step as a function of both the input as well as the prior activity. Right? And the word prior should trigger something in it, right? Yeah.
For discretize this? Because this is just a linear.
Yeah, I mean, I'm just doing it because I want to connect it to the Bayesian equation. I mean, just a standard inference equation. So here's the question you can ask, which is, can such an equation, which is a biologically motivated equation, implement Bayesian inference over time, right? So let's look at this very popular model for hidden Markov models, which is a very standard model for modeling things that change over time. So what does this model have? Basically, it has this assumption that the world can be in one of n different states at any point in time. So I'm calling that theta 1, theta 2 up to theta m. And then the assumption is that the world is changing in a Markov manner, which is that the state at time t depends only on the state at time t minus 1. So that's given by this matrix. It's called the transition matrix. And so graphically, you can look at it in this particular way, that the state is changing. It depends only on the previous state. The arrow is a dependence assumption, right? And then how does that relate to the data that you're getting? The data is generated using another this likelihood function, right? So p of I given theta is given by this arrow here. So that's basically how the world is evolving. That's the assumption you're making. That's the model that you're saying captures the evolution of data over time. And so given this particular model of how your data is being generated, you can then ask the question, what is the probability of a particular state, theta t, is equal to I or j or whatever the state is, given that I've seen everything in the past. So from time step t all the way up to time step 1, all the data I've seen so far, what is the probability distribution, the posterior probability of theta, given all the data, right? And that's the inference problem, or the filtering problem if you want to use the engineering term. Okay, so here's the equation for that, right? So what is the probability of the current state, so theta being equal to I, given that I've seen all the data from the time step 1 all the way up to time step t. So this is just Bayes' rule, right? So I'm making the assumption about conditional independence and so on. You get the posterior is equal to the likelihood, which is this part, and then the prior. This is the prediction based on all the past data. And you can simplify that by using this transition probability. So you just have a summation over all the previous states, j, of the estimate from the previous time step t minus 1. And that's basically giving you a way to update your posterior given both your current input as well as your prior from the time step t. Okay?
K is the number of steps.
So that?
What's a K? K is the number of steps.
Yeah, so K is basically just the normalization constant. If you want this to sum to 1, right, then you can basically sum out these quantities and make this the normalization constant, right? So you can also make it proportional to that, right? So if you don't like the K, you can make it proportional. But if you want to keep updating, then you have to compute the normalization constant, and it's just a K. Okay? But basically, what I want you to do here is just to take home the message that you can do Bayesian inference over time by just assuming that the current input is giving you the likelihood. So that's this I given theta at time t. And then the past estimate was this estimate here. This is exactly the same as this, except for t minus 1, right? So use that, propagate that information through time using this transition model, t given t minus 1, and sum it all over all the past states, j. And then you get this prediction, right? So this part is just the prediction of what you expect that theta to be. And then you multiply it with the likelihood. So you have the prior, you have the likelihood, and this is Bayes' rule multiplying, and you get the new posterior. And it's a recursive equation, right? So you can keep updating over time, right? And another way of looking at that is to say there's a current input influenced term here, and then there's a feedback term from the past.
I don't think the updating part. You update the time step at time t, and that's it, right?
Yeah.
Next time it is t plus 1.
Yeah, so you update t, right, and then that becomes a term here for the next time step, and this becomes t plus 1, right?
Yes, but
So,
t is never updated again.
T is never, yeah.
Yes, you're shifting this.
Yeah, so t is basically, so what you're doing here is filtering, right? Which is you're, at any point in time, you're saying what is the posterior distribution given what I've seen so far, right? And then you keep updating that. So here's the question you can ask, which is, we already have this equation, which is sort of, we said this is how neurons behave in a network of connected neurons. And we have that equation that says that's how you want to do inference if you have this Markov model of things evolving over time. But is there a way to map one to the other, right? Because that would show that you're able to do inference, right? So do you notice a similarity there? There seems to be a lot of similarity, right? There's things that depend on the past, there's a summation and so on, but what's the big difference there?
One is probabilistic, the other is
Yeah, but I mean in terms of mathematical equations, right? There's a plus there, right? So how do you turn multiplication into a summation? Yeah, right, okay. So here's if you do logs, right, then you get something that looks a little bit better, right? So you have a log likelihood there, the term that potentially could map to this term here, and you have another log term that has this summation of activities from the past and so on, right? So there's some relationship over there. The only problem, of course, is that they have a log of sums there and you have a sum of logs over here, right? So you have to do an approximation, and that's where the approximation comes in, is the network has to approximate the log of sums by the sum of weighted logs, right? And that you could do, you can actually show that you can do that. But the main idea is that neurons could potentially compute the log posterior probabilities of states given input data. And if you really want to get the posterior, you could do an exponential, and that is related to the spiking probability of the cell also.
So can't you just do something like assume that the way neurons work is that the firing rate is a product?
Yeah, so that's
right?
right. So that's another way of doing it. Is if you use a different model of neurons, then you could. So that would require a more complicated model, right? So if you have more complicated models where you assume they can even do products, some people suggested that. Then again, you can implement this kind of an equation. Right? So I'll give you a couple of examples of what this lets you do. So for example, if you have a really simple task where I'm giving you noisy data like this, where there's some bar that's oriented, but you cannot see it properly, but you want to integrate this over time, you can get a better idea of what that orientation is, right? So you could have a neuron that has, you have a network that has a set of weights that are encoding different orientations, such as the ones shown at the top. Those are the feed-forward weights. And the transition model is really simple here because we are assuming things don't change over time, so it's an identity matrix. So I equals j equals 1, so there's no dynamics going on here. Okay, it's just a simple static estimation problem. You want to estimate the orientation given a whole bunch of data. And so you get things like this, right? So the image sequence is something like this. Right? Can you see the bar there? It's pretty obvious, right? So given this kind of a noisy image sequence, can you infer the orientation? So the log likelihood in this case looks something like this. So you can see how it's jumping around a lot because of the noise, but it does have a peak, but the peak also shifts from left to right, but it's approximately the orientation of the stimulus, right? And then finally, in the network's output, you get something like this, where gradually the belief in the orientation at some particular orientation grows and grows and grows until it reaches a particular threshold, and then it will give you the output. So the output looks something like that. So here's what we just did, which is we got a response that the posterior was peaked around this particular orientation, so we could say, well, according to my belief, this is the orientation that I just saw in this really noisy image, right? Got a question? Okay. Was that time?
No, I have a question.
Yeah.
See, you said the posterior computes for network over time.
Yeah.
So if you see the brain as a computation machine, so
Yeah.
where does heuristics come in? Because heuristics
Yeah.
will kind of bypass all this.
Right. I mean, the heuristics here is more that log approximation that I said, and then also I haven't talked about how you learn the feed-forward weights or the recurrent weights and so on. So a lot of the learning will require you to have some amount of approximations there, and price and so on. So I think that's something I haven't talked about, but that's an important issue. Is how do you do the learning, and how do you get to the likelihoods and the here I was fixing it to solve this problem, or how do you do that from a data itself or through evolution, right? Here's another example, which is a very common task used to study decision making in monkeys and humans. So we can actually do this task also. So there's a bunch of dots that are moving randomly on the screen, but some fraction of them, let's say 50% or 100%, will be moving in one direction, and you have to tell me which direction these dots are moving in. It could be left or it could be right. Okay, so here's the one example, left or right? Right? How many left? Can't tell. Okay, if you're a monkey, you can tell because they've been trained so well that monkeys are really good at this. But if you're human, humans are not good at it because we haven't been trained, right? So, but monkeys can do this at the threshold of like 2-3% of the dots moving in the same direction.
It's like trained monkeys or untrained monkeys?
Trained monkeys. But here's a much easier version of the task. How about this? Pretty easy, right? So all I did was increase the coherence to 50%, and you can all tell what direction they're moving in. And so here's a network that can start to solve this problem. So in this case, you want to actually solve this motion direction problem. So each of these dots are moving locally here in this one-dimensional image, and then there's some transition probability of moving from left to right or right to left. And so you can use a network like this to solve this particular direction of motion task. The feed-forward base in this case, for example, could be just localized position, so positional inputs to the network. And then the feedback is encoding the motion direction, so you can have something like this, where each of them is encoding some constant velocity. So this matrix is telling you that given that I'm seeing something in one position, it's likely that it will move to the next position. It's a very simple, sort of almost diagonal matrix, but not really diagonal. And the weights for that is approximates this log of sums with the sum of logs, and that's shown on the right-hand side. It looks very similar also. And when you run this network on examples, here's what you get. So you see that the likelihoods are just position-based, no directional information, so you have two different peaks depending on where the motion is. But then you have this log posterior, which has a higher peak in this case, a rightward moving input. It has a higher peak in this set of neurons, which are right selective. And then if you exponentiate that, you get a nice peak for the posterior, which is close to 1. And the same thing for leftward moving input. So this is just for a bar that's moving in a particular direction. Okay, not the dots, but just one bar that's moving in one direction or the other direction. Okay, and then you can apply this to this task that we just saw. So here's the output. So here's the one-dimensional version of it. You can see that there's a very noisy output from the cells that are trying to encode the direction of motion. So you can see that there's peaks in both left and right. But you can use this now and integrate this information to solve this task. So here's here's one way of solving it. So you can integrate that information over time. And so the black one is the one that is integrating the information for leftward direction, and then the dotted one is rightward. And you can see that one of them, if it reaches a particular threshold, you can use that as a way to make a decision about which direction it is.
What is the y-axis transition then?
Which one?
The previous one.
Okay, let me show this again. I think this will have the same data. So this one?
Yeah, and the lower plot, what's the y-axis?
This is time, basically. So as time goes on, you're integrating. Oh, y-axis. Yeah, so this one is basically it's the posterior probability over the entire image. Okay? No, I mean, this convention is that one is, I'm just doing it symmetrically, so one is for leftward direction, other is for rightward direction, so I've just flipped it, actually. But it's not really supposed to be a probability there, right? So basically what I'm doing here is to convert that information, which is actually in the probability space, converting that into some decision variable. So the decision variable is accumulating that information over time, and then using that to make a decision. So in the next talk, I'll actually get into more detail into that particular aspect. But right now, just imagine that you're using the information from the network to integrate it over time to eventually get to a threshold. Okay, well, the next talk will get into that whole detail about how that happens. Okay, so here's that's another example where there was noise here, but this is a summary of that. So what you get is basically an effect of the noise, right? So the noisier you are, the longer it takes to reach a threshold. And the faster is the reaction time if you have a higher coherence, which is sort of what you expect, right? Reaction time is faster if you have less noise, okay? And I'll conclude with basically one comparison for the data. So if you look at how the monkey is solving this problem, there's a particular area in the brain called the lateral interparietal area, or LIP, and that area has neurons that basically do something like this. So the noisier the stimulus, these neurons respond according to the slope being changed according to the noise. Okay, so you can see that if it's very noisy, it takes a longer time to reach to a particular threshold. If there's less noise, it rapidly goes up to that particular threshold. And interestingly, in this case, the experimenter can predict the monkey's decision even before the monkey has made the decision. But just looking at the neuron's activity, if the neuron starts to fire and starts to go up, then before the monkey can press the button, the experimenter can say, oh yeah, this is going to be a leftward or rightward choice, right? Because it's just really, sort of like reading the monkey's mind in some sense for this particular task. But the point here is that there are examples of neural activities such as this that seem to correlate well with these kinds of models for decision making, where you're using posterior probability computations to then come up with some decision variables that lead to actual behavioral choices of the monkey. Okay. So anyway, in the talk tomorrow, I'll talk a little bit more about how this could be sort of explained, this kind of data on decision making. How can you use posterior probability distributions to actually make decisions by combining it with rewards and actions. Okay, so that'll be next time. Thanks.
Okay, questions for Rajesh? Sandeep.
So you were talking about these monkeys getting trained, and when they're trained, they can detect it even at 5% coherence.
Yeah.
So presumably, as they're getting trained, the weights and their neural networks are changing,
Yeah.
and they can do this. Does it make sense in that kind of situation from your Bayesian interpretation to ask whether what is changing? Is it the prior that's changing? Is it the likelihoods? Or is it impossible to distinguish that?
Well,
we
at
have
this,
access to the final log probably.
yeah, at this point we don't have the kind of resolution we need to really answer that question. In terms of the data that we have is usually only from one brain cell in one particular area, so you need to really target a whole bunch of areas and so on.
But in principle, you
in principle,
you
yeah.
can distinguish those. So
Yeah.
in this network
Right.
model
Mm-hmm.
of the neural network and its
Yeah.
dynamics,
Yeah.
you can separate what aspects of it affect the prior and which aspects affect the likelihood, or is it impossible to break them down?
Well, I think in the model you could do those kinds of experiments, right? But the problem is that we still don't have a good way of mapping those kinds of models, which are basically a recurrent network with feed forward and feedback and so on, to the actual data from the brain, which involves anatomical connections that are across multiple areas and so on. So you could do it for very sort of close to the sensory sensory devices, like in the retina. You could probably do it in the retina and then the nucleus that I talked about. But once you get to the other areas of the brain, like this LIP, it's already gone through a whole bunch of stages that it's very difficult to pin down where the actual changes are happening, because it's probably happening in a whole bunch of different places. So I think at this stage we're still not able to pin down the exact kind of locations of plasticity that would lead to this kind of behavior.
Jose, I'll come back.
Well, I have some questions.
Yeah.
The questions are more a matter of principle. First is, how did you write your likelihoods? Where did you get your likelihoods from?
Right, so I didn't really go into detail here, but there's a way to, there's multiple ways of doing it, but the one that I talked about here was based on a simple linear filter model. Right, so basically saying that the image multiplied by a linear filter, this is basically a vector, and it's proportional to that, so it's proportional to the response of that.
No, I saw the model you had,
Yeah.
what you call the hidden Markov
Yeah.
model. Basically, it's a Markov model.
Yeah.
It's a Kalman filter model.
Yeah, that one is, yeah. It's a,
But even in a Kalman filter model,
Yeah.
the system equation is a linear equation.
Right.
But your likelihood and your prior are doubts for the standard Kalman filter.
Right.
So, did you assume a doubt system?
No, this was a discretized model, right? So each,
Oh, so but you got your likelihood, well,
just to the equation. So if you go back to this equation here,
but I think you're doing something circular.
yeah.
You're using the data to get the likelihood. Then you're
Yeah.
computing the posterior based on the data.
So this is the likelihood, right? This part of it here. And if you look at the equation itself, the way it's doing it is by relating it to this product, right? So this W is basically a linear filter. Its different orientations are going to give you different responses for a single image, right?
But in a standard common filter,
It's not a common filter. I mean, it's not a continuous. It's a discretized version.
that's
right?
fine.
Yeah.
No, I have no problem with
Yeah.
that. But it's the basic idea is that of a common filter.
The graphical model is that of a common filter, but the implementation is not. Yeah.
But the likelihood is not specified from the data. If you're specifying the likelihood from the data, I think what you're doing is something circular, and it's like using the data to predict the same data.
No, I mean, what you're doing here is using the, so you're talking about this term here? Like how is the
I'm
likelihood
asking
given
a
by?
basic question.
Yeah.
To compute the posterior probability,
Mm-hmm.
you need the likelihood.
Yeah.
And the general strategy is that the likelihood should, the specification of the likelihood should be independent of the data. The
Right.
data tells you what the likelihood is.
Yeah.
The data fits in.
Mm-hmm.
So I guess the question is, do you have a model that gives P of I of t given
Yeah.
theta of t?
That's right, and that's,
Model given T of
that's this model here,
That-
right?
I see.
That's that model there. It's basically a linear filtering
Yes, but are
operation.
you,
Yeah.
you're learning it from a different set of data, or are you?
Yes.
Yeah, so you're talking about learning the matrix, this matrix W, right? This vector W, right? And so that's being done. I haven't shown it here, but you can do that through. People use natural images to do that. So basically, the assumption is that the brain has evolved to process natural images, so it's learning these filters based on natural statistics.
I won't pursue this matter,
Yeah.
but let me just step back a little bit more.
Yeah.
The impression I got from the title of your talk is, does the human brain act like a Bayesian machine? That means an untrained human being in the calculus of probability and Bayes' law should really do what Bayes' law would have done had it been a machine. The impression, that was my impression. The message that I'm getting is, you're analyzing brain data using Bayesian methods. That doesn't mean that the brain acts like a Bayesian machine.
Right.
A non-Bayesian could say, I could have analyzed this data too, and I could have claimed that the brain doesn't act like a Bayesian machine.
Yeah, I mean, it goes back to this question we had earlier, right? So why is that the only model that can explain the data? And like I said, there's it's not just one particular experiment, but there's a whole class of experiments that are all explained quite succinctly by making this particular Bayesian assumption. So I think it's sort of the argument of simplicity. So it goes back to this idea that before we had these kinds of models, people had these tailored models for each individual phenomena in perception in this case, or action. But now we have this one model that can explain a very diverse set of data, which is why a lot of people are attracted towards these models. And what I talked about was, if that is really the case, then the brain must be implementing some of these equations, right? Using the circuitry, the biological algorithms that it has. And so the question is, how do you bridge that gap between the mathematical abstraction and the biological implementation? And in doing so, can you relate that to the data that's being collected by recording from the brain? But the argument for the non-Bayesians, I think, comes from this, that the sort of simplicity of fitting the data with just a single model.
My question is, by using this analysis, can you discriminate between your dynamical models and estimate dynamical model parameters?
Yeah, so you're again getting into the learning part, right? So how do you actually learn? In this case, it was a hidden Markov model, so you have the likelihood as well as the so you have the observation model, and then you have the transition model. And so I didn't talk about the learning part here, but yes, there are ways in the statistical literature. There's this EM algorithm that they use to estimate that from data and so on. And there have been attempts to then show you can approximate those kinds of algorithms for these kinds of neural implementations. So there's been work done. I haven't really talked about that, but yeah, I think I can point you to the literature on that.
Okay. So I'm not a computational neuroscientist, but I've heard that in your model, the state of a sort of a cortical area is represented by the firing rates of these neurons, right? So these are firing rates.
That's right.
But apparently, people can do tasks like you can flash photographs by me, and I have to press a button whenever a photograph contains an animal picture in it, and I can do that at a rate that is really quite high relative to the rate at which firing rates change appreciably in the brain.
Right.
Arguing that there must be more than just firing rate. So can you maybe just comment on what your...
Yeah, I mean, that's a big debate. So classically, people have looked at firing rates as being the information coding entity in the brain in terms of, you measure the firing rates because the brain is very stochastic. I mean, you record from the same neuron to the same stimulus, you're going to get different patterns of spikes. And so the way to get around that is to say, okay, I'm just going to look at the firing rate and see how that changes as a function of time. Right? So basically, the more recent work has shown that you can actually make decisions like the one you said with even a few spikes. So you don't have to really calculate an entire interval, but only a few spikes. So the one way you can do that is by averaging over a pool of neurons, not just a single one, but a pool. And that gives you an approximation. So each of those neurons is computing similar set of states. Then the average can be computed over a pool. So that's one argument for how you could get firing rates, even in a very short interval, or even in a one delta interval. You can get a firing rate estimate by averaging over a group of neurons, right? But the other alternative is to say the inference is being done not with firing rates, but with spike timing. So that's another group of people who are looking at how do you do inference with spikes, single spikes, not with a firing rate-based model. And that gives you a lot of interesting ideas about sampling-based inference, so not using this kind of a discretized inference, but maybe using stochastic sampling like Monte Carlo type methods. And the brain could be doing that, right? So that's another alternative class of models.
Okay, I'm sure there'll be more questions for Rajesh. Thanks, Rajesh, for a very interesting talk.